Okay, I'm George Kidder. This is Anna Farrell. We're from the MDI Biological Lab, the Community Environmental Health Laboratory. This talk was supposed to be given by Jane Disney, who has had some family problems that she had to deal with. So Jane is a hard person to replace. It's taken two of us to do it. And we'll see if even with the two of us we can come up with anything that looks reasonable. Because you're going to be hearing a talk which was basically written by Jane and then modified by Anna and myself, and then sort of winging it from here on. Can you run the slides? Good. Let's just talk for a minute about what eelgrass is, because you know, botany is something that people tend to forget about. Eelgrass is very uniquely an angiosperm. That is, it is a flowering plant. And it is not only that, but a very unique aquatic angiosperm in that almost uniquely, there are a few other species that do this, it spends its entire life underwater, including pollination. It produces a very curious kind of pollen, which is not insect pollinated, it's not air pollinated, it drifts around in the ocean. And then it produces seeds, and it can reproduce that way. It can also reproduce by cloning. That is, it grows a rhizome which puts up shoots at various intervals, and the above ground parts, that is above the ocean sediment, are the photosynthetic parts of the plant. Now, because of this curious structure, the above ground parts have a lot of air spaces in them. That's why they float up off the bottom, because they have these air spaces. And this has some very interesting consequences. One of them is that the oxygen that they produce in the process of photosynthesis, you all remember your elementary biology course, the oxygen they produce in photosynthesis tends to stay within the blade, and because it gets into these air spaces, it diffuses rather rapidly down to the roots. Diffusion of oxygen in gas is about 1,000 times faster than the diffusion of oxygen in water. So it tends to stay within the plant, goes down to the roots. Not only does it provide oxygen for the roots, which are not photosynthetic, of course, but that's where the oxygen leaves the plant. And this reduces the anaerobic nature of the substrate they're sitting on. It tends to eliminate the hydrogen sulfide and makes it possible for other things besides the eelgrass to grow in this substrate. So an eelgrass bed, the substrate around an eelgrass bed is much less anoxic than it would be without the eelgrass. The other thing that happens is that because of these air spaces, if you rip up an eelgrass plant, as you might, for instance, if you were dragging for mussels or scallops or cockles or mussels or alive-o, if you rip up the eelgrass and then say, well, we'll just throw the eelgrass back overboard, it floats. And that means it doesn't get anchored in the bottom. This business of being anchored in the bottom is essential to the eelgrass. Okay. You don't have to tell people that any vegetation like this provides habitat for fish and other invertebrates. All you have to do is put some in your aquarium and watch the guppies swim around in it. But it also provides a place where invertebrates live on the surface that other fish eat, and other fish eat those organisms and so forth. It provides a very important part of the habitat, including, well, what have we got here? We've got a starfish growing in what looks like a mussel shell. We've got some snails growing on the living on the eelgrass beds. They're grazers. They're probably taking algae off of the eelgrass surface, and so forth. It's a very important ecological part of the world, and we've been having trouble with it. And I'll pass it over to Anna to tell us the trouble that we've been having. But eelgrass is in difficulty, and the question for this talk is, why is it in difficulty, and what can we do about it?
Okay, so there are several threats to eelgrass habitat. Invasive species is one. This is a picture of 2 green crabs mating at Wonderland this year. Jane stumbled upon them in the eelgrass bed, as she likes to say. And invasive species are disruptive to the natural environment. Shoreline erosion is another problem. It can increase turbidity in the water and reduce light penetration, reducing photosynthesis, which eelgrass needs to survive. Fishing practices such as dragging and digging and anchors and propellers can physically damage eelgrass beds. And pollution, which causes elevated nutrients, excess sediments, and perhaps oil and metal contamination, just a whole host of problems. And not up here, but equally important is climate change. This is a map of Mount Desert Island. Schoodic is the red star in the corner there. And we have seven sites we looked at this summer we were researching. Hadley Point, Berry Cove, Bar East and Bar West around Bar Island, Wonderland and Ship Harbor, and then Stave Island. And we looked at 3 different main things, and we restored at these sites. So we were looking at eelgrass abundance and green crab abundance and genetics and some water quality parameters. And we restored at all of these sites, just at Hadley Point and Berry Cove, as well as some other ones, but those are our main restoration areas this summer. And the MDI Biolab is up there as well. Okay, so this map shows eelgrass in 1996 in the Upper Bay. Just to give you where we are, this is where the Biolab is in Salisbury Cove. Berry Cove and Hadley Point are here. And the green shows healthy eelgrass beds naturally occurring. 1996, like I said, this was mapped by aerial photography and showed 3,174 acres of eelgrass. So we use 1996 as our baseline year for our restorations. And so this is 2008, same area, and you can see the green has greatly decreased. This was aerial photography again, and we have 1,128 acres of eelgrass in this area. And then in 2013, something happened and it all disappeared. So there's very little green. This little patch right here is this green dot. Hadley Point, which used to look like this, now looked like this. No grass, just some snails. So we wanted to figure out why. Where did it all go? Let me back up a little bit. So in 2007, Jane Disney started restoring eelgrass at Hadley Point. She saw it declining and saw a need to restore. So she engaged a diversity of community partners and volunteers through this process, like Maine Coast Heritage Trust, who remains a partner. This is Terrytown right here. He did a lot of boat captaining for us this summer. But the main restoration process was harvesting from a healthy eelgrass bed. This is at Stave Island. So we would gather bundles of eelgrass. And then at low tide, we would tie the eelgrass onto these 2 by 2 foot biodegradable grids and take them into the water and put them down, and the eelgrass plants would take root and grow off the grids and come up the next year in greater abundance. So it worked. At Hadley Point, after 5 years of restoration, eelgrass populations increased from less than 0.5% to 24%. So things were looking good until 2013. In addition to the restoration, Jane facilitated with the Frenchman partners conservation agreements. So this map was created in 2013. It was a live GIS mapping event. Mussel draggers and harvesters and scientists from the lab got together and decided that after talking through it, what the mussel harvesters wanted from the bay and what we were trying to do with eelgrass restoration, they decided that these areas where the red hash marks are would be no harvest zones. So in the state of Maine, you cannot restrict dragging except commercially. These are not commercial. This is just an informal agreement based on the goodwill of the participants, and it's held up, and I'll tell you why it's important in and of itself. It's one of the first agreements of its kind, but I'll tell you why it's become even more important later. So in addition to restoring this summer, we wanted to understand the loss, because if we're restoring and it's just disappearing, what's the point? So we looked at two different possible causes. We had two undergraduate interns this summer, and one looked at degradation of water quality, and the other looked at the impact of the invasive green crab. And this picture is actually from this summer. This is Stave Island, which has somewhat declining eelgrass populations, but it's still fairly healthy. So this is a really low tide. People are harvesting for a transplant later on in the week. So nice and healthy. So we looked at several different parameters of eelgrass and water quality. We looked at eelgrass abundance, eelgrass biomass, blade tensile strength, and tissue nutrient content. And for water quality, we looked at dissolved oxygen, salinity, turbidity, temperature, inorganic nitrogen, ammonium, and phosphates, so kind of general run-of-the-mill water quality parameters. And then we ran these against each other, and I'll explain some of the results. Just a note on how we calculated eelgrass abundance. We had these 25 by 25 centimeter quadrats, and we'd throw them randomly in the eelgrass patches and count the blades and the number of shoots, and then use this measuring stick to measure 3 blades at random. And one day Jane was doing this, and you kind of stir up the sediment when you're counting the shoots, and she feels something grab onto her finger. She pulls her finger up, and there's a baby lobster attached to it. So she had quite a fun time with that. And it just kind of underscores the importance of these habitats, for developing fish and shellfish. Eelgrass biomass, this is actually the day where it happened. We threw the same quadrats randomly and removed all the above ground growth and scraped the blades clean of epiphytes and such, dehydrated the sample, and then weighed it. And what we found when we plotted this, so on the x-axis we have site name. There's 6 different sites. And we have abundance over here and biomass over here. There was definite differences between sample sites. Some sites were more abundant, some sites had more abundant eelgrass, some sites had more biomass. And biomass and abundance were correlated between sites, which was expected. Patches with higher shoot counts tended to weigh more. And then we looked at tensile strength. So one of the interns made this tensometer. He would put these washers on a weight, and when the blade broke, that was the tensile strength. So tensile strength in this case was breaking strain divided by blade width, or per blade width. And the sample site, again, samples on the x-axis, tensile strength, which is in newtons per centimeter on the y. Sample site had a significant impact on tensile strength, and the intern looked at 24 blades per sample site. So when we ran all these parameters against each other, eelgrass with water, or abundance with water quality, biomass with water quality, tensile strength with water quality, we didn't really find anything that stuck out. There wasn't anything significant. So water quality variables do not have a significant impact on eelgrass health that we found. What can be taken away from this, besides that, is that measures of eelgrass health vary between sites. So biomass and abundance and tensile strength were wildly different between sites. So simultaneously, another intern was looking at the impact of the invasive green crab, which has been hyped up in the media, lots of talk surrounding the green crab. And we looked at eelgrass biomass and density, again, for this one. and then abundance and haplotype of the green crab. So there are two different haplotypes, northern and southern green crab. And we were asking the question of whether the northern green crab had spread to Mount Desert Island, and if so, did the green crab composition vary between eelgrass, vary with eelgrass biomass between sites? So there have been two invasions of the green crab in the Gulf. The southern haplotype came over from southern Europe about 100 years ago and shipped ballast water. And then in the 1980s, the northern haplotype came over from northern Europe. And some researchers in Canada suggested the northern haplotype was more aggressive. Okay, so then to determine if it was one or the other, northern haplotype or southern haplotype, we looked at the CO1 gene, which is recognized as a marker for population differences. And the intern collected 15 adult crabs, green crabs, from each site and extracted the DNA and sequenced it. There's one of the sequences at the bottom. These numbers up here represent differences in the nucleotides at different loci in the gene. Haplotype differences are displayed in relation to the H1 gene. That's just kind of an arbitrary distinction. But H1 and H3 haplotypes represent southern variety, and H4, H5, and H6 are northern haplotypes. Then we put all together what we discovered, and so we had expected that at sites where there wasn't much eelgrass, like Berry Cove and Hadley Point, hardly any at all, we would see a dominance of the northern haplotype, and this wasn't the case. Almost at all sites, the southern haplotype dominated. Bar East, which has a fairly healthy eelgrass abundance population, had the most northern green crabs. So our hypothesis that northern green crabs were the instigators of the eelgrass loss was not true. And we did prove that the northern haplotype is on Mount Desert Island, though. And this just shows that there's no correlation between average eelgrass abundance and the fraction of northern haplotype green crabs by site. So we thought, okay, if it's not the haplotypes, maybe it's just the sheer number of green crabs. So we went out and did 4 days of trapping at 7 sites on Mount Desert. These are the traps we used. This is Darius, who was very excited to do green crab trapping. And this is just one of the green crabs that we collected. We sexed and measured them all. And we collected 578 over the course of the 4 days. Let's see. Okay, so then we ran this against plant density. Density was shoots per quadrat, which we averaged, and this is average crab abundance. And there was no significant relationship there either. For example, these 2 points right here, there are 40 crabs at each, but differing plant density, so no relationship there. And we ran this against eelgrass biomass, and again, no significant relationship. This is the biomass on the x-axis and crab abundance, on the y. So, in sum, the green crab results, we hypothesized the northern variety was destroying eelgrass, and there was no correlation between green crab haplotype and eelgrass abundance. Then we thought, well, maybe the number of green crabs was correlated to eelgrass density and biomass, but again, we found that it wasn't. So while all this was going on, we scaled up our restoration efforts. We transplanted at 5 different sites this summer, from May to September. This is our tried and true method. They're the 2x2 foot grids I was talking about earlier. We made a lot of them. That's what I spent my winter doing. It was so much fun. So we had these string grids, which are the original prototype, and those are really time consuming to make beforehand during the winter. So we thought, okay, maybe we can shorten this by using burlap, and we would just weave the eelgrass plants into the burlap. That turned out to be super time consuming at the shore, which is when we can't afford to lose time, because you only have so much time on the low tide before you're in over your head. So we decided, okay, maybe we can move away from grids, and we tried washers. So we would tie plants onto the washers, drop them over the boat. We weren't constrained by the tides this way, but we didn't like the thought of putting all that steel in the ocean, so we tried rocks. So we just went along the shore and got rocks and tied eelgrass to that. And I think we're not completely satisfied with that, so we're going to try some other things next summer. And these are two of our young environmental leaders. Jane runs a program for students during the summer, and they saw these extra strips of burlap, and they thought, oh, we could do something with this. So they created these burlap runners where they wove the eelgrass in and tied some sandbags on to weigh them down, and then kind of unrolled them in the ocean, like the red carpet. And those worked okay. We did a couple of them. They didn't stay down very well. You know, the water currents tended to push them up and twist them around. So we've been trying out a lot of different things. But in total, we transplanted 8,060 eelgrass plants this summer. And while we were transplanting, we discovered late in the summer, this is at Berry Cove, but also at Hadley Point, eelgrass was coming up from seed. So these seeds have been dormant for 2 years. We didn't think that eelgrass could germinate after 2 years of dormancy, but it clearly is. It's small, it's patchy, but it's there, which is really important. It's coming up in places where eelgrass was naturally originally and also where we restored, so we know both methods are important to eelgrass coming up from seed. And this is why agreements like the one the Frenchman partners made with mussel harvesters are super important. If these areas had been dragged while the eelgrass seed was dormant and just uprooted all that, it wouldn't be coming up. So even when there's nothing coming up, it's important to conserve and preserve these areas. So in conclusion, it's not excess nutrients, it's not salinity, it's not dissolved oxygen, it's not turbidity, it's not entirely the green crabs. It is related, I'm talking about the loss of eelgrass here, some fishing practices, maybe herbicides, definitely climate change in some capacity. So this is by no means an exhaustive list of possibilities, but merely the beginning of an effort to understand eelgrass loss in Frenchman Bay and bay dynamics. So thank you.
All right, we have time for a few questions. A couple, all right.
Thank you. Can you put Frenchman Bay eelgrass in a larger context in terms of what's being seen elsewhere?
In the Maine context, there is eelgrass, or has been historically eelgrass, at a whole variety of sites up and down the eastern coast. Casco Bay had large amounts of eelgrass. Most of the work which has been done south of here indicates that the loss of eelgrass that was seen was probably due to nutrient pollution from land-based sources, which caused an overgrowth of algae, which cut down the amount of light, which knocked out the eelgrass. It certainly was true in Great Bay in New Hampshire. It probably was also true in Casco Bay. That makes it a very much more difficult problem for them than for us, because our water quality has not changed. North of here, the losses seem to have been green crab driven. Although, as you see, when you try to quantitate that, it doesn't seem to work out very well. Worldwide, there are species of eelgrass, not only this particular species, but there's some 20 others, and they're found essentially all over the world. I'm not sure if I've answered your question properly.
Great. Other questions? I saw a couple more. One over here.
I really enjoyed the South Park-esque video. And I'm wondering if things like that and the kind of hype that you talked about with green crabs and their impact, despite the fact that you have, at least in the ways that you've looked, the green crabs don't seem to be correlated with losses of eelgrass abundance and density or biomass. I'm wondering if that kind of media or hype is potentially problematic to trying to explore other options and talk to people about exploring those other options. Are people convinced it's the green crabs and they don't want to hear anything more about it because of stuff like that? Though I loved it.
I think you put your finger on it. As a retreaded laboratory physiologist, one of the things that really disturbs me about this whole field is the difficulty of obtaining hard data. And you have to remember that this study that we did with green crabs was done in a year following a green crab crash, if you want to call it that. For the past 2 years, the number of green crabs you could catch in the bay has been going up and up and up. This last summer, there were times when we wanted to get green crabs for the people in the lab to work. We couldn't find the green crabs they were looking for. In other words, a cold winter knocked the green crabs way back. So we're studying eelgrass, which may be strongly affected by things that happened 2 years ago against the population of green crabs that's there now. I wish we could have started back before we thought green crabs were a problem, but you never think of doing those experiments. I gather we're out of time.
Yes.
Okay.
Wonderful. Well, thank you very much, Anna and George.